Good afternoon, dear students. Lecture number 7. The topic of the lecture: Hereditary and Multifactorial Human Diseases. Plan of the lecture: Hereditary diseases classification, gene diseases causes, metabolism diseases general signs examples, and the next one: chromosomal aberrations causes clinical manifestations, diseases with an unconventional type of inheritance, and in conclusion, multifactorial diseases. All diseases, depending on their development, can be divided into 3 groups from a genetic point of view: hereditary diseases, diseases from hereditary predisposition, not hereditary diseases. Hereditary diseases are diseases that are caused by mutation leading to change in genetic material. There are 4 options of the hereditary diseases. Gene diseases, in which changes in the structure of the DNA molecule occur. Chromosomal mutations that disrupt the structure of chromosomes. And genomic mutations, resulting in changes in the number of chromosomes. Well, let's speak about all these diseases. So, gene diseases. Gene diseases are a diverse group of diseases caused by mutations at the gene level. As you know and remember, there are different mutation types: missense, nonsense, sameness, for example. Replacement of the nitrogen bases led to formation of a stop codon. A nonsense mutation occurs and the shortened polypeptide chain forms. The number of gene diseases in humans is really high. Already more than 3,500 gene diseases are inherited according to Mendel laws, according to 3 main types of inheritance: autosomal dominant, autosomal recessive, and sex-linked dominant and recessive. You see the pathogenesis of a gene disease on the screen. Mutant allele, pathological primary product or pathological mutating protein, chain of biochemical processes of cells in organs and organism. A large group of gene diseases with an elucidated biochemical defect are enzymopathy. Enzymopathy are metabolic diseases associated with the reduction or complete suppression of enzyme activity. Molecular diseases are called. The enzymopathy have several general signs. At first, clinically manifest in homozygotes. And the next one, in heterozygous carriers of the mutant gene, enzyme activity may be somewhat reduced. And the next one, in the heterozygote, insufficient enzyme activity can be detected by biochemical method. Inherited by autosomal recessive or X chromosome-bound recessive type. Loss of activity of enzymes led to blockade on the way of conversion of one substance to another. The accumulated substances may have a toxic effect or have a toxic effect on the produced byproducts. The metabolic diseases include hereditary disorders of metabolism of carbohydrates, amino acids, lipids, and etc. For example, Galactosemia. Galactosemia is disorder of carbohydrates metabolism. Disease development is associated with a mutation in a GAL gene, encoding the synthesis of the enzyme galactose-1 phosphaturidine transferase. It catalyzes one step of converting galactose to glucose. This mutation leads to insufficient or complete loss of enzyme activity. The disease forms after birth when lactose from mother's milk begins to enter in newborn body. The clinical symptoms are diarrhea, vomiting, jaundice, increasing weight loss, late developing mental retardation. The treatment occurs with the help of elimination of lactose from the diet. The next gene diseases are amino acidopathy or amino acid exchange disorders. They have the common features shifting the pH of the medium to the acid side and bringing out the corresponding amino acid or product of the metabolism with urine. The most studied and widespread diseases of this group include phenylketonuria, tyrosinnemia, albinism, and ketonuria. All these diseases are related to each other by shared metabolic paths of amino acids. We will speak about phenylketonuria now. The disease is associated with the failure of the enzyme phenylalanine 4-hydroxylase, mutation in the gene of race, conversion of amino acid phenylalanine to tyrosine is disturbed. As a result, the toxic phenylpyruvic acid forms. It is accumulated in different tissues, including the nervous tissue. This leads to violation of the functions of organs and systems. A child with phenylketonuria is born healthy, but in the first week of life, if he is breastfed, symptoms of the diseases develop. Increased excitability, spastic muscle tension, convulsive epileptic surges. Later, the mental retardation, reduced pigmentation of the skin, hair, and iris develops. The treatment is elimination of phenylalanine from the diet. The next point of the lecture is chromosomal anomalies. There are about 700 chromosomal mutations have been described. Approximately 40% of spontaneous abortions are caused by a chromosomal imbalance. For every 1,000 newborns, there are 3 to 4 chromosomal diseases. Structure of chromosomal anomalies are anomalies of sexual chromosomes, 35.7%, trisomies by autosomes, 24.5%, unbalanced chromosomal structural change, 4.9%, and balanced structural change, 34.9%. Chromosome mutations include two groups. Mutations resulting in excess of a portion of the material of chromosomes. For example, duplication, unbalanced translocation. And mutation leading to a lack of part of genetic material on a given chromosome. Deletion, deficiency, and translocation. For example, Cat's-Cry syndrome. This is the deletion of fifth chromosome, short shoulder. The frequency of its occurrence is 1 on 45,000 newborns. The symptoms of this disease are low birth weight, lack in mental and physical development, microcephaly, moon-shaped face, hypotension of the muscles, and a kind of crying that resembles the meowing of a cat. The life expectancy of such patients is usually small. Death occurs from pronounced disorders of the internal organs. The next group of diseases is genomic mutations. The genomic mutations or change in the number of chromosomes are the reason of monosomies and trisomies. For example, Patau syndrome is trisomy of 13th chromosome. Edwards syndrome is trisomy of 18th chromosome. Down syndrome is trisomy of 21st chromosome. Cytogenetic forms of Down syndrome are in 93% of patients, trisomy 21st chromosomes. 5% have translocation of 21st chromosomes, and 2% have a mosaicism. The frequency of its occurrence is 1 to 2 on 1,000 newborns. The clinical traits of Down syndrome are mental retardation, microcephaly, deformed ears, thick lips, thickened tongue, flat back of the nose, excess skin on the neck, joint instability, transverse palm line, and monkey fold. And there are abnormalities of organs development, heart defects, kidney hypoplasia, esophageal and duodenal stenosis. The next point of the lecture is diseases with the unconventional type of inheritance. These diseases include mitochondrial diseases, diseases of a genomic imprinting, and diseases of expansion of trinucleotide repeats. The first group is mitochondrial diseases. The reason is mutations in gene encoding tRNA, rRNA, oxidative phosphorylation proteins. The deletion nucleotide insertion in mitochondrial DNA occur. I think you remember that mitochondrial diseases are inherited only on the mother's side. The most common mitochondrial diseases are on the screen. For example, Leber's hereditary optical neuroretinopathy, Kearns-Sayre syndrome, Pearson syndrome, mitochondrial myopathy syndrome, encephalopathy, lactic acid acidosis, stroke-like episodes, among others. Let's speak about Leber's hereditary optical neuroretinopathy, for example. The reason of this disease is the replacement of guanine with adenine at the same position of the ND4 gene. It leads to a severe loss of vision. On the screen you see the image of normal optic nerve and atrophy of an optic nerve. The next group of diseases with an unconventional type is diseases of a genomic imprinting. Genomic imprinting is an epigenetic mechanism of regulation of homologous genes during the development of the body, depending on the parental origin of the gene, chromosome, and genome. Cytosine methylation occurs, which results in a transcription block. The most common diseases of imprinting are Willis syndrome, family hypertrophic cardiomyopathy, spina bifida, epilepsy, Prader-Willi syndrome, Engelmann syndrome. For example, Willis syndrome. The reason is the deletion of the long arm region of the 7th chromosome, about 26 genes. The frequency of its occurrence is 1 on 20,000 newborns. The symptoms are airy face, narrow chest, low waist, elongated neck, and etc. Children lag behind in growth. There is a delay in mental and physical development. In most cases, cardiovascular diseases, aortic stenosis, lesions of the gastrointestinal tract, and urological disorders are associated. The next group is diseases of trinucleotide repetition expansion. The reason is the repeated repetition, expansion of repetitions of CAG, CGG, and CAA triplets. These diseases have some common properties. They are on the screen. At first, dominant type of inheritance. Secondly, the disease begins when the number of repeats reaches a certain magnitude. And the next one, there is a phenomenon of anticipation, an increase in the severity of clinical manifestations of the disease in subsequent generations. In the next one, the existence of a clear adhesion of the mutant allele to a certain chromosome has been proven, indicating the existence of the founder effect when such a mutation occurs and remains in the population. For example, Huntington, chorea. The reason is a mutation in the IT15 gene, leading to a repeat of the CAG triplet more than 36 times. As a result of this change, the Huntington protein accumulates inside the neural nuclei in the striatum of the brain, which leads to the degeneration. When Huntington is broken down, toxic products are formed that cause the death of nerve cells. The average age of onset of symptoms is 41 years. The average duration of the disease is 15 years. Gradually, there are mental disorders, decreased intelligence, dementia. The frequency of occurrence is 1 on 10,000 or 1 on 20,000. The next point of the lecture is mitochondrial diseases. These are diseases caused by a combination of hereditary predisposition and external factors. A group of diseases differs in nosological forms among cardiovascular, nervous, mental, skin, eye, child, and others. For example, ischemic heart disease. This is the acute or chronic heart disease resulting from reduced or discontinued myocardial blood supply due to a pathological process in the coronary artery system. Risk factors for this disease development you see on the screen. Genetically determined: gender, physique type, coronary vascular structure, lipid metabolism disorder, blood clotting system disorders, arterial hypertension, and etc. And environment factors of these diseases: smoking, unbalanced diet, low mobility, exposure to negative psychological and psychosocial factors. The next example of multifactorial diseases is diabetes mellitus. Risk factors for diabetes mellitus are: the first group, genetically determined: atherosclerosis, arterial hypertension, excess body weight, and environment factors: unbalanced nutrition, low mobility lifestyle, systematic alcohol consumption, frequent infections, inflammatory diseases. And the next example of multifactorial disease is bronchial asthma. The bronchial asthma has two groups of etiological factors. Candidate gene are T and B cell receptor genes, genes of factors of inflammation, bronchial smooth muscle cells receptor genes, xenobiotic biotransformation genes, and etc. Provoking factors of development and exacerbation. Inductors, allergens, medicinal household food, and triggers, factors of escalation initiation. Cold air, dust, heavy smoke, smoking, professional aerosols, bacterial infection, emotional stress. Thus, the facts accumulated by medical genetics to date show the diversity of the correlative role of heredity and environment in the development of different types of pathology. Therefore, future doctors need knowledge of various factors and mechanisms of the occurrence of diseases in order to diagnose and prevent hereditary and multifactorial diseases. Thank you for your attention.